All right, so we start with the anatomy, the organs of the male reproductive system, where, as we've said already, primary sex organs, the gonads in the male system, are the testes. In the testes, sperm cells or spermatozoa develop, and male sex hormones are synthesized. The other structures of the male reproductive system are termed as accessory sex organs or secondary sex organs. They are responsible for transporting the sperm cells and their secretions to the outside of the male body and to secrete male sex hormones. These accessory sex organs include some that are internal and others that are external. So the first of our illustrations here, let me see if I can get this positioned well, with a side view here. Of course, here's the urinary bladder. Let's see where to start. A good place, we'll start down here with the testis, which is encased in the scrotum, which I don't think you get to see. All right, move that up a little bit. So the scrotum is the skin that surrounds the testis. This convoluted tubule here is called the epididymis, and that courses its way down along the backside of a testis and makes a sharp U-turn, and as it ascends up into the body, it is known at that point as a ductus deferens, or more commonly, I think you would probably be familiar with the term a vas deferens. A vasectomy cuts that so that the sperm produced here can't make their way out. You see that the vas deferens passes over the pubic bone and comes up and over around behind the bladder, and back here behind the bladder it joins with this gland, which is a seminal vesicle, and both those tubes come together as the ejaculatory duct, and that joins with the urethra, which of course is the exit from the urinary bladder, and the urethra then passes out through the penis to the external urethral orifice. And the yellow right in here is the prostate gland. There's that label. So we've already seen this in the section about the urinary system. This section here of the urethra in males is called the prostatic urethra because it is surrounded by the prostate gland. And then right here is the urogenital diaphragm. This has the sphincter muscles that allow you to hold it when you need to pee. And also joining into the urethra right in this area, this little gland right here, is the bulbourethral gland, bulbourethral, also known as the Cowper's gland. And let's see what else needs to be pointed out. So the penis has 3 columns of erectile tissue in it. There are 2 side by side on what's considered the ventral side that are called corpus. Well, one is a corpus cavernosum. The plural form of that is corpora cavernosa. And then there is one corpus spongiosum. Now, the corpus spongiosum extends down to the end of the penis and forms the glands, what most people refer to as the head. And that courses back up into the body and expands on the inside and forms what's referred to as the bulb, B-U-L-B, the bulb of the penis. The corpora cavernosa, you don't see it here, but they split out on both sides and are attached to the pubic bone. I think we'll see that in the next illustration. So here's a different view. Testis, epididymis, vas deferens, joining to the seminal vesicles, kind of on the back and underside of the bladder, passing through the prostate gland. Then You have the bulbourethral glands here on either side of the urethra, right below the prostate gland. And then the urethra passes through the purplish colored corpus spongiosum. And then on either side of that are the two corpora cavernosa. Now, this part here they're referring to as the root of the penis. You see how the corpus cavernosum kind of splits off. And what that does is that's what's attached to the pubic bone. All right. Let's see now. The testes, which is the plural form of the word, singular is testis. Of course, we're also familiar with the terms testicles. Ovoid, which means egg-shaped structures, about 5 centimeters long, 3 centimeters in diameter. Both testes are suspended from the trunk by a spermatic cord and are found in the cavity of the sac-like structure called the scrotum. In a male fetus, the testes originate from masses of tissue behind the parietal peritoneum near the kidneys. And typically a month or two before the child is born, the testes descend to the lower abdominal cavity and pass through the abdominal wall into the scrotum. There's a male sex hormone, perhaps the main one, called testosterone, that is being secreted by the developing testes. It stimulates this descent. And then there is the fibrous cord that we saw in the opening of the chapter called the gubernaculum, attached to each of the developing testes and extends into the inguinal or groin region of the abdominal cavity. Passes through the abdominal wall, fastened to the skin of the scrotum. As the body grows, the testis remains anchored to the gubernaculum, and so the testes are guided by the gubernaculum and descends down through the inguinal canal in the groin and enters the scrotum. Each testis carries a developing ductus deferens, some blood vessels, and some nerves. And later in development, that ductus deferens, along with the blood vessels and the nerves, forms part of the spermatic cord that the testes hang by. As the testis descends, there's a pouch of peritoneum called the vaginal process moving through the inguinal canal into the scrotum. In about 25% of males, the pouch stays open, which provides a potential passageway through which the intestines can bulge through and produce an indirect inguinal hernia. And in this situation, the intestines go through that opening that the testis pass through on its way to the scrotum and can actually cause a bulge on the lower abdomen and can be problematic. The intestines can become choked by squeezing through that small opening, have to be put back into place. If it does get pinched there and the blood supply is cut off to that section of intestine, then it's called a strangulated hernia. Now, if testes don't descend into the scrotum, they cannot produce sperm because the temperature in the abdominal cavity is too high. We'll see a little bit more about that later, but that is why the testes are in the scrotum like they are instead of up inside the pelvis, the abdominopelvic cavity, like the ovaries of females are. That's because sperm production requires a temperature that is slightly lower than body temperature. The condition of undescended testes is called cryptorchidism, and if untreated, the cells that normally produce sperm cells will degenerate, causing infertility. All right, as far as testis structure, there's a tough white fibrous capsule called the tunica albuginea. Okay, remember, albu or alb means white, and this is made of a white connective tissue. Along the capsule's posterior border, the connective tissue thickens and extends into the organ to form a mass called the mediastinum testis. And from there, layers of connective tissue called septa pass into the testis and divide it into sections. So these septa are like little walls, and they subdivide each testis into about 250 sections called lobules. Each lobule has anywhere from 1 to 4 coiled, convoluted seminiferous tubules. Each of those are about 70 centimeters long if they were uncoiled. So they're highly coiled and compacted. Now, 70 centimeters, you think about that, that's a little over 2 feet long. And if you have 4 of those, that's 8 feet in each lobule times 250 lobules. We're talking about a couple of thousand feet. And then double that for 2 testes. So nearly a mile of tubes folded up tightly and compact to fit inside 2 testes. These tubules course posteriorly, in other words, they move toward the backside and unite and form a network of channels called the reedy testis, which is in the mediastinum testis. That gives rise to several ducts that join a tube called the epididymis. The epididymis is coiled up on the outer surface of the testis, and it continues on and becomes the ductus deferens or vas deferens. Not sure how clear that is in this drawing. It's kind of small, but again, the tunica albuginea, and we will see that term again. You know, a tunic is a surrounding, a wrap, and it is made of white fibrous connective tissue. So we're going to see that term again in a couple of other organs here. But divided up into sections called lobules, 1 to 4 seminiferous tubules in each one. They join together at the reedy testis. That leads to the epididymis, and that turns around and becomes vas deferens or ductus deferens. And the spermatic cord up here, you see that in that you have the vas deferens, you've got blood vessels, you've got nerves. Okay. Let's see here. The internal accessory organs of the male system. First is the epididymis. Narrow, tightly coiled tube, about 6 meters long if uncoiled. Now, 6 meters is about 20 feet. Each epididymis is connected to the ducts in the testis. Emerging from the top of the testis, it descends along the posterior surface and then courses upward. In other words, turns around and heads up and becomes ductus deferens. Interlining of the epididymis is a pseudostratified columnar epithelial cells, with long microvilli called stereocilia. These cells secrete glycogen and other substances that support the stored sperm cells and promote their maturation. Now here's a cross section, so you can kind of see the stereocilia here, and you can see some of the sperm cells in here as well. It probably needs to be magnified a little bit more for that to be clear. There, it's a little more obvious. Sperm cells and then the stereocilia. Now remember, cilia are hair-like structures, and their movement will create a flow in the fluid to move the sperm cells along. When immature sperm cells reach the epididymis, they are non-motile. In other words, they don't move. But as they travel through the epididymis as a result of rhythmic peristaltic contractions, they mature, and after the aging process, they can move independently and are able to fertilize oocytes. They usually do not move independently, though, until after they have been ejaculated out of the body. The ductus deferens, also known as the vas deferens, a muscular tube about 45 centimeters long, lined with pseudostratified ciliated columnar epithelium. They originate at the lower end of the epididymis. They pass up along the medial side of the testis, become part of the spermatic cord, then passes through the inguinal canal, enters the abdominal cavity outside the parietal peritoneum, courses up over the brim of the pelvis, extends back and toward the middle into the pelvic cavity, and ends right below the urinary bladder. And if you take a look at this, you notice that the lumen is irregularly shaped. It's not round, and that there is a thick layer, relatively speaking, of smooth muscle. So it's a very small lumen surrounded by a good bit of muscle to move the sperm along, push them along with peristaltic action. Near its termination, in other words, where it's about to end, the ductus deferens dilates into a portion called an ampulla, and then just outside the prostate gland, it becomes slender again and unites with the duct from the seminal vesicle. And where those two come together, we have what's termed the ejaculatory duct. Now that passes through the prostate gland and joins to the urethra. The seminal vesicles, also known as seminal glands, convoluted sac-like structures about 5 centimeters long, a couple of inches. Each is attached to a vas deferens or ductus deferens on the posterior surface near the base of the bladder. The glandular tissue that lines the inner wall of the seminal vesicles secretes a slightly alkaline fluid, which will help regulate the pH of the tubular contents as the sperm cells travel to the outside. Now remember, sperm cells are alive, so they are going to be creating energy. They would have to have energy for their flagella, their tail, to be able to move, for them to swim. And as a result, they're going to be producing carbon dioxide and water, which means they're going to be producing carbonic acid. And so that acid needs to be neutralized, and that is the point of the alkalinity of the secretions of the seminal vesicles. Also, it says that the seminal fluid neutralizes the acidic secretions in the vagina to help sustain the sperm cells that actually enter a female reproductive tract. Seminal vesicle secretions also include fructose, which is a monosaccharide, a sugar, to provide energy for the sperm cells. And there are hormones called prostaglandins. In fact, those hormones, and we talked about them way back in chapter 13 briefly, and they got their name because this was where they were first found, was in prostate secretions. Those prostaglandins help to stimulate muscle contractions in the female reproductive organs. They will cause the uterus to contract, which again creates somewhat of a peristaltic type motion to move the sperm up through the uterus toward the uterine tubes, which is where the sperm are actually going to meet up with the egg. Secretion of the seminal vesicles contributes about 70% of the volume of semen. Let's see. As the sperm move through the ductus deferens into the ejaculatory duct, contents of the seminal vesicles empty into the ejaculatory duct to increase the volume of fluid discharge from the ductus deferens. Then we have the prostate gland. Again, prostate, not prostrate. There is no R in that word. Chestnut-shaped structure, about 4 centimeters across, 3 centimeters thick, surrounding the proximal part of the urethra. Again, that's called the prostatic urethra. It also wraps around the ejaculatory ducts just below the bladder. It has many branched tubular glands enclosed in a fibrous connective tissue capsule. Then there are septa of connective tissue and smooth muscle that separate the glands, divide them up into sections. The ducts of these glands open into the urethra. The prostate releases its secretions into the urethra as smooth muscle contractions in its capsular wall. As this release occurs, contents of the ductus deferens and seminal vesicles also enter the urethra to increase the volume of the fluid. Secretions of the prostate gland, it's a thin, milky fluid. It includes citrate, which acts as a nutrient for the sperm, and prostate-specific antigen, or PSA, which is an enzyme that helps to liquefy the semen during or following ejaculation. Prostate gland is small in boys, begins to grow in early adolescence, reaches adult size several years later. Usually doesn't grow anymore until age of around 50, when in about half of all men, and it enlarges enough to press on the urethra. And this can cause a feeling of urge to urinate frequently because the bladder can't empty completely. If anybody watched the old cartoon show King of the Hill, Hank Hill would say, "I have a restricted urethra." That was a poor imitation of Hank Hill, sorry. All right, then we have the bulbourethral glands, also known as Cowper glands. Two small structures, each only about a centimeter in diameter. It's like the tip of your pinky finger. They're located inferior to the prostate gland and on the sides of the intermediate part of the urethra, which again is called the membranous urethra. They are enclosed by the muscle fibers of the urogenital diaphragm. They are composed of many tubes with epithelial linings that secrete a mucus-like fluid. This is released in response to sexual stimulation to help lubricate the tip of the penis in preparation for intercourse. Then we'll take a look at the semen itself. The fluid that the urethra conveys to the outside during ejaculation is called semen. It consists of sperm cells that came from the testes. It has the secretions from the seminal vesicles, the prostate gland, and the bulbourethral glands. The pH is about 7.5, just a little bit on the alkaline side. Again, that neutralizes the acidic environment of the male urethra and the female vagina to help protect the sperm. And then there are those hormone-like substances called prostaglandins, and the nutrients that we've discussed are also found in the semen. Volume of semen released at one time varies from 2 to 5 milliliters. Average number of sperm cells is about 120 million per milliliter. Now that's kind of mind-boggling to me, because I know you've often heard it only takes one sperm cell to fertilize an egg. At 120 million times, let's say 5 milliliters, go the upper end of the range, that's 600 million potential children to be produced. And there are only about 350 million people in the United States. So now we know now that not every sperm cell is really intended to fertilize an egg. It takes, as we'll see, this comes up actually in the next chapter, but it really takes a team effort of several sperm for one to be able to penetrate into an egg cell to fertilize it. So you have, it almost seems wasteful, but it's a necessary thing. The sperm cells don't move. They are non-motile, while in the ducts of the testis and epididymis, they begin to swim as they mix with the secretions from the accessory glands. They can't fertilize an oocyte until they enter the female reproductive tract. Duh. Here they undergo capacitation, which weakens the acrosomal membranes. We'll see what an acrosome is in the next section. When sperm cells are placed with oocytes in a lab dish to achieve fertilization, which is called in vitro fertilization, chemicals are added to simulate the capacitation. Sperm cells can live for many weeks in the ducts of the male reproductive tract, but usually survive only up to 6 days after being expelled or ejaculated out, even if maintained at body temperature. The ability of a sperm cell to fertilize an oocyte will only last 24 to 48 hours after ejaculation. But they can be stored and kept viable for years if frozen at temperatures below 100 degrees Celsius. And interestingly, when frozen that cold and thawed out after years, they are ready to go, ready to swim again. Okay, let me see. I believe. Okay, here we go. The external male accessory reproductive organs. First, the scrotum, which of course is the pouch of skin with subcutaneous tissue hanging from the lower abdominal region, posterior to the penis. Subcutaneous tissue lacks fat. It has a layer of smooth muscle that constitutes what's called the dartos muscle, and this is for temperature control. Remember that I mentioned a little while ago, sperm can't be produced at normal body temperature of 37 Celsius, 98.6. This dartos muscle can contract or relax depending on temperature. So that will relax to allow the scrotum to hang a little more loosely, to get the testicles a little further from the body so that they'll be a little bit cooler, 3 degrees Celsius, around 5 degrees Fahrenheit below body temperature. So that is important for sperm production and survival. It's even said that men that wear tight underwear can go sterile or have fertility problems because of temperature. There is a wall called a medial septum that divides the scrotum into 2 chambers with a testis in each one. Each chamber has a serous membrane covering the front and the sides of the testis and the epididymis. Then the penis, cylindrical organ that conveys both urine and semen through the urethra to the outside, and is specialized to enlarge and stiffen, which enables it to enter a vagina during intercourse. That's something we see here in a few minutes, called, of course, an erection. The body, or the shaft of the penis, has 3 columns of erectile tissue, a pair of dorsally located corpora cavernosa, and a single ventral corpus spongiosum. There is a tough capsule of white dense connective tissue called, I told you we'd see this again, the tunica albuginea that surrounds each column. Then there's skin and subcutaneous connective tissue with abundant smooth muscle. So if we do a cross section here, the 2 purple regions are the corpora cavernosa, and you notice there's an artery right in the center of those. That's going to deliver blood, and what these are designed to do is inflate with blood, fill up, and that produces the erection. And as they fill with blood, the vein that allows the blood out kind of gets compressed so that the blood can't flow out as fast as it flows in. And you also have the corpus spongiosum. There's only one of those. And at the distal end, this expands and forms the glans, or head of the penis. And you also notice that the urethra runs through the middle of the corpus spongiosum. Here on this side, we've got skin, subcutaneous tissue, connective tissue, the external urethral orifice at the tip, of course the glands. Now here they're showing the prepuce, which is sometimes known as the foreskin. Of course, foreskin is often removed surgically in a procedure called a circumcision. I saw an article the other day that said for the first time in U.S. history, the number of uncircumcised males exceeds the number of circumcised ones in the U.S. So the corpus spongiosum, which surrounds the urethra, enlarges at the distal end and forms the cone-shaped glans penis that covers the ends of the corpora cavernosa and also has the urethral opening called the external urethral orifice. And they mention the loose fold of skin called the prepuce or foreskin. And they talk about it being removed in the procedure called the circumcision. And the root of the penis, the columns of erectile tissue separate. The corpora cavernosa diverge, which means they split apart and spread out toward the sides, laterally again, meaning toward the side, in the perineum, and they are firmly attached to the inferior, the underside surface of the pubic arch with connective tissue. These parts form what's called the crura of the penis, and the single corpus spongiosum enlarges between the crura as the bulb of the penis, and that's attached to membranes in the perineum. Don't know if we've talked about the perineum. That's the space between the scrotum and the anus. All right, erection, orgasm, ejaculation, during sexual stimulation, parasympathetic impulses, okay, not sympathetic, not fight or flight, got to be relaxed. From the sacral part of the spinal cord, release a vasodilator called nitric oxide. Its chemical symbol is NO. It dilates the arteries that lead into the penis so that more blood flows into the erectile tissues. Increasing pressure of the arterial blood entering the spaces of the erectile tissue will compress the veins. I mentioned this a few minutes ago, to reduce flow of venous blood away from the penis. So the blood accumulates in the erectile tissues. The penis swells and elongates to produce an erection. Culmination of sexual stimulation is an orgasm, which they describe as a pleasurable feeling of physiological and psychological release. And in males, orgasm is accompanied by emission and ejaculation. So let's look at the distinction here. Emission is movement of sperm cells from the testes and secretions from the prostate and the seminal vesicles into the urethra, where they're mixed together to form the semen. It's a response to sympathetic impulses from the spinal cord that stimulate peristaltic contractions in the walls of the testicular ducts, the epididymis, and the ejaculatory ducts. So in other words, this is taking all the parts necessary for semen, bringing them together, mixing them together, loading them to the ejaculatory duct. And then ejaculation is let's just read the whole thing here. As the urethra fills with semen, sensory impulses are stimulated past the sacral part of the spinal cord. Motor impulses are conducted from the spinal cord to certain skeletal muscles at the base of the erectile columns to cause them to contract. This movement increases the pressure in the erectile tissues and aids in, here it is, forcing the semen through the urethra to the outside of the male body, which is ejaculation. Let's see. I believe that is all we have for this section. And it is. Still a fairly long video. I apologize, but just didn't seem like a good way to break that up.